Unveiling the ingenious strategy of cancer, harnessing neighboring cells for its advantage. Imagine a cunning mastermind who manipulates its surroundings to gain power and control. That's precisely what cancer cells do, according to groundbreaking research conducted by ETH Zurich scientists. Led by Professor Sabine Werner, the team discovered that cancer cells can infiltrate healthy neighboring cells, transforming them into accomplices that aid in the tumor's growth and survival. This revelation not only deepens our understanding of cancer's tactics, but also opens doors to potential therapeutic interventions. But here's where it gets controversial. Could this discovery lead to treatments that target not just cancer cells, but also the innocent bystanders they manipulate? Cancer's covert operation. Smuggling mitochondria into healthy cells. At the heart of this strategy lies the mitochondria, the cell's powerhouse responsible for energy production. Cancer cells, it turns out, can stealthily transfer their mitochondria into nearby connective tissue cells, specifically fibroblasts, through minuscule tubular bridges formed from cell membrane material. This process, akin to a sophisticated smuggling operation, reprograms the fibroblasts, turning them into tumor-associated fibroblasts, TAFs. These TAFs then undergo functional changes that significantly benefit the cancer cells. The transformation of fibroblasts into cancer allies. Once reprogrammed, TAFs exhibit altered behavior, multiplying at a faster rate and secreting growth factors and cytokines in abundance. They also increase the production of adenosine triphosphate, ATP, the energy currency of the cell. This enhanced activity fuels the proliferation of cancer cells, making tumors more aggressive. Moreover, TAFs modify the extracellular matrix, the network that supports tissue structure and influences cell behavior. By increasing the production of specific matrix components, they create an environment conducive to cancer cell growth and survival. A chance discovery leads to a potential therapeutic target. The initial observation of these tubular connections between fibroblasts and skin cancer cells was serendipitous, as Werner accounted. Her former postdoctoral researcher, Michael Cangrama, stumbled upon these nano-connections during an experiment involving co-cultures of fibroblasts and skin cancer cells. Further investigation revealed that cancer cells exploit a mechanism typically used for cellular repair, turning it against the body to facilitate their own expansion. This finding adds a new dimension to our knowledge of cancer biology, as previous studies had shown that cells from the tumor microenvironment can transfer mitochondria to cancer cells, enhancing their fitness. However, the reverse transfer from cancer cells to healthy fibroblasts was previously unknown. MIRO2, the protein facilitating mitochondrial transfer. To unravel the molecular underpinnings of this mitochondrial transfer, the researchers identified a protein called MIRO2. This protein is abundantly present in cancer cells, capable of transferring mitochondria. The team observed high levels of MIRO2 in both cell cultures and human tissue samples, particularly in invasive tumor cells located at the tumor's edge and in close proximity to fibroblasts. The detection of MIRO2 in these specific areas confirmed its role in facilitating mitochondrial transfer. Targeting MIRO2, a promising pathway for cancer therapy. The discovery of MIRO2's involvement in mitochondrial transfer offers a potential avenue for therapeutic intervention. When the researchers inhibited the production of MIRO2, the transfer of mitochondria was effectively blocked, preventing the transformation of fibroblasts into TAFs. This strategy proved successful in both in vitro experiments and mouse models. However, translating this approach into clinical applications requires identifying an inhibitor for MIRO2 that minimizes adverse effects in humans. While the development of such a therapy may take years, the potential impact on cancer treatment is significant. In conclusion, expanding our arsenal against cancer, The revelation of cancer cells' ability to reprogram healthy neighboring cells by transferring mitochondria sheds light on the complex dynamics within tumors. This insight not only advances our understanding of cancer biology, but also highlights potential targets for therapeutic strategies. By targeting proteins like MyRO2, researchers aim to disrupt the intricate web of interactions that cancer cells exploit for their survival and growth. As we continue to unravel the mysteries of cancer, the hope is to develop more effective treatments that can one day eradicate this formidable foe. And this is the part most people miss: the profound implications of targeting not just cancer cells themselves, but also the mechanisms they use to manipulate their surroundings.